In this lecture, we will discuss about the urea cycle. So, before discussing the main topic urea cycle, first we all need to know about the urea. What is urea? Urea is the end product of protein metabolism. Always keep it in mind that the end product of protein metabolism is urea. The next very important point about urea is that it is the major disposal form of amino group derived from amino acid. So, why we are discussing this topic urea cycle? Why this topic is so important? So, urea cycle is literally very important because as we all know, the nitrogen of amino acid converted into ammonia, and ammonia is very toxic for our body. So, that ammonia, with the help of this urea cycle, we can convert that toxic ammonia into urea, and urea is non-toxic in nature. So, that's why urea cycle is very important for our body or for us. Now, come to the main topic, that is urea cycle. Urea cycle is also known as Krebs-Henselt cycle. तो अगर एग्जाम में आए क्वेश्चन एक्सप्लेन Krebs-Henselt cycle, तो you will explain the urea cycle only. Now the urea cycle occur in the liver of our body, and the initial step of urea cycle occur in the mitochondria, and the remaining steps will occur in the cytosol of the cell. So, start from the first step. The first step of urea cycle is the condensation of ammonia and carbon dioxide. When ammonia and carbon dioxide condense, it results in the formation of carbamyl phosphate. Here 2 ATP is used. It is also written here 2 ATP is converted into 2 ADP. The very important enzyme, that is carbamyl phosphate synthetase 1 enzyme, is used in this reaction. So, it is written here carbamyl phosphate synthetase 1. So, it is written 1 here, तो कहीं ना कहीं इसका 2 भी होगा. तो when you will study the de novo synthesis of pyrimidine, then carbamyl phosphate synthetase enzyme is used there. So, carbamyl phosphate synthetase 1 is used in urea cycle, and carbamyl phosphate synthetase 2 is used in the de novo synthesis of pyrimidine. So, don't confuse between first and second. So, the first step is the condensation of carbon dioxide with ammonia, and carbamyl phosphate is formed with the help of enzyme carbamyl phosphate synthetase 1, and 2 ATP is used here in this reaction. Now, the next step is the combining of carbamyl phosphate with ornithine, and citrulline is formed in this reaction. Now, this citrulline, it enter from mitochondria to cytosol. So, it cross the mitochondrial membrane and enter into the cytosol, and in cytosol this citrulline condense with aspartate. When citrulline condense with aspartate, it results in the formation of arginosuccinate. So, arginosuccinate is formed in this reaction. So, the enzyme is used here is arginosuccinate synthetase, and in this reaction it is written here 1 ATP is converted into 1 AMP. It means 2 high energy phosphate bond are used here in this reaction. So, the next what was the step is the citrulline enter from the mitochondria to cytosol, and it condensed with aspartate, and when citrulline condensed with aspartate, it results in the formation of arginosuccinate, and arginosuccinate synthetase enzyme is used here in this reaction, and 1 ATP is converted into 1 AMP, which means 2 high energy phosphate bonds are used in this reaction. Now, the next step is the when arginosuccinase enzyme, this arginosuccinase enzyme cleaves the arginosuccinate into arginine and fumarate. So, what is the function of arginosuccinase enzyme? It cleaves the arginosuccinate. Here it is written arginosuccinate. So, arginosuccinase enzyme cleaves the arginosuccinate into fumarate and arginine. Now, this fumarate removes from the urea cycle, and arginine, the next step is that the arginine break down into urea. This arginine break down into urea as well as ornithine. Urea, it is the main product of urea cycle. and next is the ornithine, which again enter into the mitochondria and condense with carbamyl phosphate, and again citrulline is formed, and so on. So the end product is the urea cycle, or which is the major product also. So the overall reaction is the condensation of carbon dioxide with ammonia. After that, carbamyl phosphate is formed, which combined with ornithine. Then citrulline is formed. Then citrulline cross the mitochondrial membrane and enter into the cytosol and condense with aspartate. The next is arginosuccinate is formed. After that, arginosuccinase enzyme cleave arginosuccinate into arginine and fumarate. Fumarate removes from the urea cycle, and arginine again break down into urea as well as ornithine. So urea is formed in this cycle. So this is the process, or this is the cycle with the help of which we can convert the toxic ammonia into urea. So this is the urea cycle. Now there is a very important point here. Why this aspartate is entering into the urea cycle, and why this fumarate is removing or releasing from urea cycle? What is the significance? So this is really very important point. When you will see the structure of urea, this is the structure of urea. When you will see the structure of urea, then there are two amino groups present in the structure of urea. First amino group and the second amino group. Now the question is that these two amino groups comes from where? ये amino group जो urea cycle में है, ये हमें कहाँ से मिले? So the first amino group comes from the ammonia. Your answer, the first answer will be first amino group comes from ammonia. This ammonia, and the next amino group comes from aspartate. When you will see the structure of aspartate, this amino group is present in the structure of aspartate. Okay, so first ammonia, so there are two amino group present in the structure of urea. So first amino group comes from ammonia, and the next amino group comes from aspartate. So aspartate enter into urea cycle and it donates its amino group, and this remaining carbon skeleton is known as fumarate. When see the difference, the only difference is the amino group. Aspartate have contain amino group, whereas fumarate do not have amino group in their structure. So aspartate enter into urea cycle and donate its amino group, and the remaining carbon skeleton is fumarate, and this fumarate removes from the urea cycle. So this is the importance of aspartate. So this is the very important and common viva point of view question कि how many amino group present in the urea in the structure of urea. So you will answer there are two amino group present in the structure of urea. And the next question, examiner का next question होता है कि ये amino group कहाँ से आए, कहाँ से मिले? यूरिया में जो आए दोनों अमाइनो ग्रुप, ये कहाँ से आए? तो what you will answer? The first amino group comes from ammonia, and the next amino group comes from the aspartate. Okay. Now the next is the regulation of urea cycle. The regulatory enzyme of urea cycle is carbamyl phosphate synthetase 1. So carbamyl phosphate synthetase 1 is the first enzyme in urea cycle. It is the regulatory enzyme also of urea cycle. So examiner will ask, or anyone will ask, what is the regulatory enzyme of urea cycle? So what you will answer? The regulatory enzyme of urea cycle is carbamyl phosphate synthetase 1. Okay. Now the next is a very important point is that there is a activator. There is an activator of carbamyl phosphate synthetase 1, and the name of that activator is NAG, N-A-G, NAG. The full form of NAG is N-acetyl glutamate. So what is N-acetyl glutamate? N-acetyl glutamate is an activator of carbamyl phosphate synthetase 1. So what is carbamyl phosphate synthetase 1? It is the regulatory enzyme of urea cycle. So the activator of CPS1, carbamyl phosphate synthetase 1, is NAG, N-acetyl glutamate. This N-acetyl glutamate is formed with the help of acetyl CoA and glutamate. Here, when acetyl-CoA condense or combine with glutamate, it results in the formation of N-acetyl glutamate (NAG), and CoA will release with the help of enzyme N-acetyl glutamate synthase. Now the next is the energetics of urea cycle. We have already discussed that in the first step 2 ATP was used, and in third step 1 ATP was converted into 1 AMP. It means 2 high energy bond phosphate bond were used here, and 2 ATP were used here. So we can say that in urea cycle consumes 4 high energy phosphate bonds, 2 from first step and 2 from third step. So your how many ATP or high energy phosphate bond consume in urea cycle? So you will answer 4 high energy phosphate bond consume in urea cycle. Now the next is the disorders of urea cycle. As we all know, there are total 5 enzymes present in urea cycle or used in urea cycle. So due to the deficiency of these 5 enzymes, there are 5 disorders of urea cycle. So the first enzyme was carbamyl phosphate synthetase 1. So due to the deficiency of enzyme carbamyl phosphate synthetase 1, the disorder will be so due to the deficiency of first enzyme carbamyl phosphate synthetase 1, the deficiency disorder will be hyperammonemia type 1. The next enzyme was ornithine transcarbamylase. So due to the deficiency of this enzyme, the disorder will be hyperammonemia type 2. Next was the third enzyme was arginosuccinate synthetase. So due to the deficiency of enzyme arginosuccinate synthetase, citrullinemia will occur. The fourth enzyme was arginosuccinate lyase. It is also known as arginosuccinase. हम लोगों ने जो यूरिया साइकिल में पढ़ा था वो आर्जिनोसक्सिनेज लिखा था, तो डोंट कंफ्यूज बिटवीन आर्जिनोसक्सिनेज एंड आर्जिनोसक्सिनेट लाइएज, बोथ आर सेम. सो ड्यू टू द डेफिशिएंसी ऑफ दिस एंजाइम द डिसऑर्डर विल बी arginosuccinic aciduria. The last enzyme was arginase. So due to the deficiency of enzyme arginase, the disorder will be arginemia. Okay. Now in the third column it is written product accumulated. This is really a very important column as well as very easy. तो अगर आप लोगों ने यूरिया साइकिल अगर आपको आती है बहुत अच्छे से एकदम क्लियर है कि सारे स्टेप के सारे एंजाइम्स वगैरह सब क्लियर हैं, तो यू विल राइट द नेम ऑफ प्रोडक्ट दैट विल बी एक्यूमुलेटेड इन विच डिसऑर्डर, कि किस डिसऑर्डर में कौन सा प्रोडक्ट हमारा एक्यूमुलेट हो रहा है. तो कोई ऐसे रटने की चीज़ नहीं है. अगर आपको यूरिया साइकिल आती है तो यू विल राइट ईजिली. तो इन द डेफिशिएंसी हाँ ओके, तो स्टार्ट फ्रॉम हेयर. कार्बामाइल फॉस्फेट सिंथेटेज 1 एंजाइम इज डिफेक्ट. कार्बामाइल फॉस्फेट सिंथेटेज 1 एंजाइम डिफेक्ट है, तो हमारा क्या होगा कि अमोनिया, दिस अमोनिया विल नॉट कन्वर्टेड इनटू कार्बामाइल फॉस्फेट. व्हेन दिस एंजाइम इज डिफेक्टेड, अगर ये एंजाइम डिफेक्ट है तो हमारा अमोनिया क्या होगा कि ये हमारा कन्वर्ट नहीं होगा कार्बामाइल फॉस्फेट में. सो दिस अमोनिया विल एक्यूमुलेट इन आवर बॉडी. So the accumulated product in hyperammonemia type 1 will be ammonia. Now the next is the ornithine transcarbamylase. If this enzyme is defect, due to the deficiency of this enzyme ornithine transcarbamylase, अगर ये एंजाइम डिफेक्ट होगा तो हमारा ऑर्निथिन और कार्बामाइल फॉस्फेट कंडेंस होकर सिट्रुलिन नहीं बनाएंगे. जब नहीं बनाएंगे तो हमारा इनका एक्यूमुलेशन होगा, प्लस साथ में हमारा क्या होगा, अमोनिया का भी एक्यूमुलेशन होगा. तो इन hyperammonemia type 2 which occur due to the deficiency of enzyme ornithine transcarbamylase, the accumulation of ammonia will take place. That's why it is known as hyperammonemia type 2. Now the third one is arginosuccinate synthetase enzyme. So due to the deficiency of this enzyme arginosuccinate synthetase, when this enzyme is defected, due to the deficiency of this enzyme citrulline will not converted into arginosuccinate, so the accumulation of citrulline will occur. Okay, the fourth one is the arginosuccinate. So due to the deficiency of this enzyme, arginosuccinate will not convert it into arginine, so the accumulation of arginosuccinate will occur. The last one is the arginase. When due to the deficiency of arginase enzyme, arginine will not convert it into ornithine as well as urea, so the accumulation of arginine will takes place in the last fifth arginemia disorder of urea cycle. This is the end of the urea cycle. Thank you and have a good day.